So, in this lecture, I would like to explain basic fundamental properties of enzyme. So, basically, in a biology, what is enzyme? Or you can say, in every reaction, catabolic reaction, there is some reaction reaction goes on the body. So, first definition of enzyme is enzyme are basic properties of reactivity, metabolic reactivity, or we can say it can increase the rate of a chemical reaction in the system of the plant and animal. So basically, first definition goes like this: enzyme are nothing but proteins, or we can say enzyme are called biological molecule or biomolecule which act as a catalyst. Catalyst is defined as substances that increase the rate of chemical reaction without being itself change in the process. It means when the reaction is taking place, when reactant is going on the process to form a product, then the catalyst play a major role to increase the chemical reaction, right? And next definition of enzymes is it can basically increase the rate of a chemical reaction by lowering the energy of activation, or we can say activation energy. And simple definition for activation energy is called activation energy is nothing but the reaction process, means it can initiate the reaction, right? And we can say that enzyme act as a catalyst. If we plot the graph between enzyme of energy with reaction process, we can get a difference of energy. That is difference of energy, nothing but called and activation energy, right? Next is enzyme have a unique three-dimensional shape of that fit the shape of reactants. And we just say that it is a three-dimensional shape. If we see the structure of enzyme, it's a 3D shape, right? Next properties of an enzyme is very important. Of this second one is that it acts as catalytic efficiency, means it has high efficiency. It ranges from 10 to the power 3 to 10 to the power 17, right? And it acts as a physiological pH at optimum temperature at normal 37 degrees centigrade temperature. Next comes our chemical composition of animal. We just see the just physical properties of an enzyme. Now goes to chemical composition of an enzyme. We just see that how enzyme act in a process. Now enzyme is classified into two parts, that is called simple protein enzyme or conjugated protein and enzyme. And if we see the structure of chemical composition of a simple enzyme, it is made up of only one type of amino acid. The simple protein is the protein or enzyme which is made of only one type of a amino acid. Next comes conjugated protein plus prosthetic group, we can say that conjugated proteins, right? If we say the hollow enzyme is the combination of apoenzyme plus cofactor enzyme, cofactor, right? Prosthetic group and coenzyme both can considered as cofactor. Firstly, coenzyme is bounded with loosely enzyme or non-covalent bond, and prosthetic group is very tight or even covalently bound to enzyme, covalent bound with tightly even covalent bound. And we see that roles of a cofactor in enzyme activity. We just see that if enzyme and substrate is both are attached to each other, then the product is formed, that is called enzyme substrate complex. And cofactor change conformation of active site, and cofactor participate in temporal bonding between active site and substrate site. It means cofactor is basically it's take participate in temporarily bonding with active site of an enzyme, or we can say and substrate site of an enzyme. And classification enzyme is basically on the basis of their composition. First is monomeric enzyme, second is oligomeric enzyme, then third is multi-enzyme complex, such as fatty acid synthesis. We know that how we can named by in a chemistry we just give a name of an compound, or we can say we give IUPAC name for the particular species. Just like here we just give a nomenclature, or we can say enzyme are usually named according to the reaction that carry out. If we see the enzyme at prefix parts and suffix parts, we just see that every last part of uh, enzyme we just say as is replaced by that particular words. Like to generate the name of an enzyme, the suffix as is added to the name of its substrate. Like lactose, os is replaced by ase, and fructose, os is replaced by ase, fructase, right? For example, we can see that DNA polymerase as a enzyme. And now we just see that what is enzyme and its classification. Now we just see the its work. It's something called function. First main function of an enzyme is its lower its activation energy. All chemical reactions have an energy barrier called activation energy, separating the reactant and product. Means activation energy is nothing but the energy that's separated from reactant and product, right? Now we just see the enzyme lower reaction activity. Here we can see the example, and we just see the role of lactose, how it breaks down into glucose plus galactose. Yeah, we can see in the carbohydrate how the lactose is break down into glucose plus galactose. Yeah, these are called epimers. We just discuss in a next lecture. Now what is the difference between enzyme and protein? We just heard that the word that enzyme is nothing but protein, but what actually difference between enzyme and protein? Here all enzyme are protein except some RNAs. Not all proteins are enzyme. We just see that all enzyme are proteins, means all the enzyme we just encountered that all enzyme are called protein, but except RNA. RNA is not an enzyme. We just see the exceptional of a enzyme. Now we just see the enzyme is a type of molecule. It has site where substrate is bind, that is called active site of an enzyme where substrate is bind. Now we just see the structure of an enzyme. Now here definition is enzyme binds substrate to the active site and stabilize the transition state of the reaction. The active site of an enzyme is the place where the substrate bind and at which catalyst occur. We just see that how enzyme plus substrate is bind and the reactant gives product like enzyme and substrate complex, right? Here we just see the structure of a enzyme. Here these follow two rules. One is called induced field theory, next is called lock and key theory. We will discuss in a next lecture what is induced field theory and lock and key theory. Here we see that this is called active site where substrate is actually bind. When substrate is and enzyme is bind together, we give enzyme substrate complex. After on we get enzyme plus product, right? Here is basic definition. Substrate approach active site. We know that substrate is approaching its active site. Enzyme substrate complex form we just see here, substrate transfer into product, yeah, product release and enzyme recycle. Now next is enzyme activity. Enzyme are never expressed in terms of their concentration but are expressed only in activity. We just see na concentration like molarity, normality, molality. We express in this into a terms called mole, or we can say microgram, milligram. We just see the SI unit, but enzyme actually does not have units, but expressed on only as activities. Enzyme activities like moles of substrate converted to product per unit time. Enzyme activities, we can say the basic definition is 1 moles of substrate that is converted to product per unit time in second. This is called enzyme activity. Next is the rate of appearance of a product or the rate of disappearance of substance. We just see in the chemical kinetics in chemistry in class 12 that chemical is reaction takes place when rate of formation of product is equals to rate of dispersion or disappearance of reactant. Now next topic is what are the factors which affect the enzyme activity, means which can affect the activity of rate of enzyme. One is concentration of substrate, concentration of enzyme, temperature, pH, activator and inhibitor. These are the fundamental things which can enhance, not enhance, it can affect the enzyme activity. If we increase the pH, then it can change the activity of enzyme. Last one is called enzyme in clinical diagnosis. An enzyme test is a blood test or urine test that measures level of certain enzyme texts. How will the body system are functioning, whether there has been tissue damage. Why? Common enzyme used for clinical diagnosis include, some of the common clinical diagnosis include alanine aminotransferase, also called glutamine pyruvate transferase, alkaline phosphatase, amylase. Amylase is an enzyme that is found in that is sucrose, not sucrose, that is called sucrose is converted into cellulose. That's called amylase. Lactate dehydrogenase. This is lactate dehydrogenase is found in a TCA cycle called trans acetylase, or we can say TCA cycle. We can also call Krebs cycle. You just see the function of lactate dehydrogenase and enzyme. So here we just know the things, what are the basics things, like what is the main function of an enzyme and what is the role of an enzyme and how it's work and what is the basics properties we just see in a enzyme, right? So here my lecture is over.